hearing from Dr. Abhay Satoskar from the Ohio State University Wexner Medical Center, presenting his research on pathology and vector-borne disease, as well as Dr. Rafael Vieira from the University of Paraná in Brazil, presenting his work on ongoing studies on ticks and tick-borne diseases in southern Brazil and Somalia, a One Health approach. With that, I will go ahead and hand it over to you, Dr. Satoskar, and you may go ahead and share your screen.
Can you see it?
Yes.
Okay.
If you could go ahead and put it into, perfect. Right?
Okay, so let's get started here. So thank you very much for having me. It's been a while since I talked on one of these, but let's give it a shot. I know we have 30 minutes, so really what I will do is I will just review what we have been doing for the last 10 years. And the title for my talk is second generation leishmanization vaccine with markerless attenuated Leishmania major strain using CRISPR gene editing. So it's basically a story where we think we have a vaccine for leishmaniasis, and this vaccine will be undergoing a phase 1 clinical trial starting 2021-22. We have all the resources in place, and this talk will basically take you through the journey of this vaccine, how it was developed. And it's an example of how folk medicine, really, leishmanization, has been translated into a realistic vaccine for this disease. So just to give you a global perspective, it's a disease endemic in all continents. Almost 10% of the world population, affects about 2 million people, which actually get the clinical disease, but infection is much higher, maybe 12 million people. It's responsible for more than 50,000 deaths, I would say around 100,000. And after malaria, it has the second highest number of DALYs, which is index of disease morbidity. There is problem because the current drugs are less than ideal. They're toxic because they need prolonged administration and not well tolerated. And even some drugs are not approved in US, like antimony by FDA. And there is no vaccine and there is no sterile cure. However, we know that people who get the disease and cure it or get treated develop a very good natural immunity, which means we should be able to develop a vaccine. So we wanted to develop a vaccine that is safe but efficacious as leishmanization, because there have been many efforts during the last 2 or 3 decades, and multiple different types of vaccines were tried, and none of them worked. And the reason we decided to take up this challenge, because of course there are no vaccines available for leishmaniasis against humans. And second most important thing is that we know that an acquired immunity develops after resolution of infection, means that you should be able to develop a vaccine. That's a basic principle of immunology, that if you get a natural immunity after, acquired immunity after natural infection, then you have in good shape to get a vaccine. If your acquired immunity is not good, you are unlikely to get a vaccine. You'll still get it, but efficacy will be lower. So as I said before, there are many vaccine strategies that were tried. Killed vaccines, recombinant antigens with adjuvants, DNA vaccines, but none of them worked, and many of them, and some of them at least, failed in clinical trials. So we decided to go back to the drawing board and see, what is the best way. And then if you go to the literature, there is a process called leishmanization, which is almost like a folk medicine, which has been there for nearly 100 years, maybe, in many countries, mainly in Middle East, where people know that if they inoculate themselves with a low dose of this Leishmania major, which is a species of Leishmania, which causes self-resolving infection in most people, almost 90% people. And that infection, if you do that, then you develop an immunity against both cutaneous and visceral form of disease. So it's almost like what they would do with smallpox. So obviously this is a virulent Leishmania major, which is not going to cause problem in majority, but still it is a wild type parasite. So of course it is not acceptable way of using it clinically, because that the regulators will have a lot of issues with this. And this strategy was used in Middle East for many years. Even now it has been used, not officially or approved, but people still do it. And many people, like 2 million people, were vaccinated in Iran during the Iraq-Iran war, in Israel and Uzbekistan, and it works. But as I said, you cannot use it because FDA will never approve a wild-type parasite. So what we decided to do was we decided to give it a thought, and then thought that if you genetically modify this live attenuated Leishmania in a way that it provides immunity without causing clinical disease, then you have a vaccine. And that was the basic premise for this project. And as we know, there are a lot of live vaccines that are safe and effective. We still use smallpox, yellow fever, polio, MMR. So if you do it right, you should have a good vaccine. And before I go any forward, this has been an effort of a team of international investigators, as you can see on this slide. Of course, Ohio State University, which is leading the project. Then you have US FDA, Dr. Nakasi's group from McGill, Dr. Matla Shivaski, who pioneered CRISPR in the Leishmania system. Then we have Jesus Valenzuela and Shadin from NIH, who have the sandfly transmission model. Dr. Hamano from Nagasaki, who is expert in preclinical characterization. Then we have a industry partner to make the vaccine. And we have Johns Hopkins now on board for helping us with its clinical trial. We have independent vaccine consultants like Dr. John Donnelly, and then we have folks in Getner Ali and others in Ethiopia for phase 1 clinical trial, which we plan to do in endemic site. And then of course, we have sites in India for phase 2 and 3 clinical trials. And we are doing some zoonotic, actually vaccine studies in dogs in Pasteur Institute in Tunis. So there's a really project is a testimony to an effort of a team of international investigators. So many of these are responsible for all these results, what I'm going to tell you today. It's easy for me to say results in 30 minutes, but there are a lot of efforts which went in, and it's not my work alone. So everyone has contributed. With that, we decide to attenuate Leishmania major. We decide to delete this gene called centrine gene. And centrine is a calcium binding cytoskeletal protein found in all eukaryotes. And it's important for duplication, segregation of centrosome. And so basically it's important for cell division. And many years back, Dr. Nakasi from FDA had found that this particular gene is expressed in amastigote stage, the stage which is pathogenic, the stage which is in the host. And they also found that this centrine is absolutely essential for replication or proliferation of these amastigotes in the host, but not for the promastigotes or the stage in the sandfly or the promastigotes which we culture in the tissue, in a culture medium. So they did that by knocking down the gene in another Leishmania, which is Leishmania donovani, and what they found was that these parasites can grow perfectly fine in the tissue culture, but when you infect the mice, infect the cells or animals, they persist for some time. In animals, they persist up to 8 to 9 months, in cells for a few more days, but then eventually they start dying. So they don't persist long, or they don't cause chronic disease. And that was the whole idea, that this could be a very nice target to make a vaccine. And we decided to do that. And the reason we decided to, the question is, why do L major? We already had donovani, and there were two problems. First, that we went to FDA, and then they said that the Leishmania donovani is a visceralizing species of Leishmania. So even if you attenuate that species and you think it is safe, when you do clinical trials, how would you monitor the dissemination of this parasite to liver? Because it is potentially visceralizing species. So perhaps in some people it goes to spleen early. And then how do you know that? And that's important because if that happens, you need to do early intervention for treatment. And the answer is you cannot. You only know when the patient develops a disease and splenomegaly, which may be too late. And the second issue was that when they knock down this gene, they use traditional homologous recombination technique. And when you do that, yes, you have a knockout, but then you are left with the parasite, which has the antibiotic selection marker gene, like neomycin or bleomycin, whatever you put it. And regulators like FDA would tell us that we cannot allow you to put any microorganism with an antibiotic selection marker gene into people. because that's again a big no-no. So we had two problems. First, that it was a visceralizing species, and second, it had the selection marker gene. So what we decided to do at that stage, what my thinking was that we know leishmanization work. We know it is caused by L major, which is mainly dermotropic species. We also know that it cross-protects against cutaneous and visceral. So if we can knock down centrin gene in Leishmania major in a way that we can also get rid of the selection marker gene, then you may have a vaccine, which is like a leishmanization. That's why we call a second generation leishmanization, but basically not causing lesions, but providing you immunity. And that's what we decided to do. And the way we decided to do was to use a CRISPR technology, because that was the best approach to delete the gene, as well as then do the single clone, single cell selection. And then after that, you could basically get rid of the plasmid with antibiotics. And we can go into details later. And to make a long story short, we got the parasites, which is shown here, which is nicely seen here. This is a centrin gene knockout. There's no centrin gene. You can see by Western blot, same thing. There is a knockout, no centrin. Wild type, there is a centrin, and this is actually a Leishmania centrin, major centrin, which we add back centrin, and it starts re-expressing. We did the full genome sequencing, and CRISPR works like a magic. We did not find even a single point mutation anywhere else. Only thing which was missing was a CRISPR, the centrin gene. In fact, what you see here is that the knockout parasites are resistant. They get some lesions, which is inflammation, but then they basically don't cause disease. This is a wild type. If you add back, it causes disease. Then we cannot detect parasite when it's a knockout, but when you add back centrin, it causes disease. So basically showing us that by deleting centrin gene by CRISPR in Leishmania major, we had an antibiotic selection marker-free centrin gene deficient Leishmania major, which was avirulent and did not cause clinical disease. So the next thing what FDA wants to know is, are these parasites safe, even before putting into any animals or humans? And we did multiple different ways to look at this. First thing we did was to see if these parasites can cause disease in severely host. And for that, we use a variety of different mice. We took interferon gamma knockout mice, rag knockout mice, which do not have T and B cells, and STAT1 knockout mice, which basically are functionally deficient in interferon response. And in all three groups, when we inject the parasite, the wild type parasite, they cause disease. If you inject the knockout parasite, they did not cause disease. As you can clearly see here, we could not see any parasites. In STAT1, we did see some parasites, but we wanted to see how much is the window, so that these mice were actually infected with 200 million parasites. Our normal dose, which is about million, so these are like 200 times more parasites. And even with that, we did not see much disease, which means that the safety window is pretty good for this vaccine. The second thing we did was that we took the mice and we inoculated them with the vaccine, and then we immunosuppressed them with dexamethasone to see if any parasites are there and they come back. And again, you can see here in the lymph nodes and the infected ear, most of the mice, even after immunosuppression, do not have parasites. 1 or 2 mice had parasites, which is okay, because that tells you that these parasites do persist up to 8-9 months, which is necessary for you to generate an immune response. But again, there is no clinical disease, as you can see here, even after immunosuppression. So this year and this year is pretty normal. But there are some parasites which will persist, which is pretty good. The third thing you want to know is these parasites basically safe in the sandfly vector? Because we know that parasites can be taken up by sandfly from a vaccinated person, and they can mix with a wild type population to give hybrids. So what we did was we inoculated mice with either wild type parasite or centrin knockout parasite, and then we feed them on an uninfected sandflies. And then we check how many sandflies are infected at 2 weeks and 8 weeks. And as you can see from this graph, the sandflies which were fed on a mice infected with wild type, majority of these sandflies are infected. However, the sandflies fed on the inoculation site where we injected L major centrin knockout, none of those sandflies are infected, indicating that these parasites cannot survive in a sandfly host, which means they are environmentally safe, which means that they're unlikely to form hybrids with the wild type parasites. Then we wanted to look at the efficacy. And this is a very important thing to remember, that a lot of these vaccines probably failed because they did not use right preclinical animal models. The easiest model to use in a vaccine will be to vaccinate an animal and then challenge them with a parasite through needle challenge. And many models work like that, but that's not the right way, because sandfly infection is quite different than a needle challenge, because sandfly saliva has a lot of different things. It has a microbiome and different molecules. They promote infection. So we decide to actually test our vaccine using the most stringent model, which is the sandfly transmitted leishmaniasis, where we immunize the mice intradermally with single dose with no adjuvant. 7 weeks later, we actually expose these mice to the bites of major infected sandflies. And after the 17 weeks after that, we euthanize and we look at different parameters. And nice, you can see here that naive mice, this is a normal ear. This is a non-immunized mice, but challenged. This ear is almost destroyed, a lot of pathology. And this is an immunized mice, which were challenged. The ear is pretty much compatible. If you look at the parasite numbers, there are about 100 parasites here. This is almost 10 to 6 at 10 weeks post-challenge. And if you follow these mice longer, these parasites will be even lower. And this is with Leishmania major, which is the homologous challenge. Now we did the same experiment where we immunized with Leishmania major, which is our vaccine candidate. But this challenge with Leishmania mexicana, which is a newer cutaneous leishmaniasis causing species, and we find the same results. These are 4 mice, non-vaccinated big lesions. These are the mice which are vaccinated, they are normal ears, which tells us that this vaccine does cross-protect against cutaneous leishmaniasis, not only in the old world, but also new world. The next important and most important thing perhaps is a visceral leishmaniasis, because that's the disease which kills and potentially fatal. And here we decide to use the hamster model. Again, we decide to use a hamster model where we infect the vaccinate the hamsters with the L major centrin vaccine, and after that we expose them to the bites of sandflies infected with Leishmania donovani. This is a gold standard model, because if you do that, this is a pretty long experiment, and if you follow the hamsters for 9 to 14 months, these hamsters will start developing disease just like humans, with weight loss, ascites, splenomegaly, and eventually they will start dying just like humans without any treatment. That doesn't happen in mouse. Mouse models, mice don't die because of L. donovani, they can control. And that's why I emphasize that it is very essential to use the right model for testing. So we did that, and I will just show you the results. This is a hamster which is non-immunized and challenged. You can see the hamster is cachectic by 9 months, losing weight. This is a hamster which is immunized and challenged, no weight loss, pretty much healthy. Next parameter we looked was the spleen, which is a pretty good indicator for severity of the disease. This is the normal spleen in a hamster. This is the spleen in a non-immunized hamster, which is the top group, the massive splenomegaly. That is what you will see in humans. And that's exactly what happens. There is no splenomegaly with immunized challenge hamsters. We can see that basically these spleens are comparable in size to these two. What about the mortality? We followed this up to 14 months, and immunized hamsters all survived. Non-immunized, everyone died. By 14 months, we had 100% mortality. If you look at the parasite loads in the organs, this is spleen and liver, you can nicely see that this parasite loads drop in the immunized. They don't, they start increasing in the non-immunized. The same thing happens in the liver. The parasite loads drop, and that's what it is, So, and last thing was to see if we can now grow these parasites in a bioreactor, and this is actually showing that we can. This is a preparation train, and you can clearly see now these parasites start from a frozen stock and then moving into bioreactor, and they are pretty healthy. So we can grow parasites up to 40 million per mil, which is pretty good because our dose is 1 million. So we can, within a mil, we can grow about 40 doses. And this is the same thing. If you scale it up to 5 liter bioreactor scale, 5 liter bioreactor scale, then also it is pretty much comparable, the growth pattern, as you can see nicely. And this is the vaccine, which is in Genoa, producing GLP conditions, and this is in the wild. This is still being tested. And so we have now optimized the conditions to make this in GLP, and those who can be easily moved to now GMP. That's what we will be doing for clinical trials. And this is what we estimate. So if we have a 1 liter bioreactor, we can have almost, 40,000 doses from 2 runs, and basically 2,500 doses from 1 lot. If you have 40 liter, we have 0.1 million doses from 1 lot. That's a lot. So basically, that is good news because that reduces the cost of this vaccine tremendously. It would be, I would say, less than $3 or $4. And that's something which is important for implementing this in the field. So in summary, we have the vaccine now. It is safe, it is immunogenic, and it is inducing immune response similar to leishmanization. And it also induces protective biomarkers in human cells. I haven't shown you the data. It is efficacious against visceral and cutaneous. And we have manufacturing process has been optimized. Analytical assays are completed on a bioreactor product. RCB prepared under CGMP. Assay development for lot release and testing is ongoing, and we are now moving forward with the preparation and characterization of master cell bank and working cell bank with BioRay Alliance USA. So with those many people, I think there will be huge global impact. This will be potential for first ever live parasitic vaccine to be licensed. Will help advancing VL elimination goals, will help controlling zoonotic reservoir if it's effective in dogs because no treatments exist. And it will be a pan-Leishmania species vaccine because it will be effective against multiple species and perhaps could be used as a therapeutic vaccine for PKDL. So I think with that, my time is done and I will stop there. Thank you.
Thank you so much, Dr. Satoskar. We have time for maybe one question if you're able to take one.
Sure.
All right. If you could just raise your hand or send a message in chat. All right, we have a message from Zalalam.
Okay.
You can go ahead, Zalalam, or I'll unmute you.
All right. Thank you, Laura. Thank you, Dr. Satoskar, for a great presentation. My question is on this centromere knockout. You just commented, you showed us the pathologies in rabbits have significantly decreased, but the graph shows the animal model still have some sort of parasite replicating in them until 9 months. Will this, my understanding, and you also commented like the sandflies are not able to replicate this centromere knockout, so that they don't cause a hybrid, like there might not be a revertant. Would that revertant be possible in the mammalian model?
No, so David Sachs and a lot of people have done extensive studies, and basically what they have shown, and there are a couple of things. So first of all, whatever happens mainly happens in the sandfly. And secondly, I haven't shown you the data, but we have passaged this parasite through animal model at least 7 times and sequenced every isolate, and we haven't seen anything changing.
Okay.
And that has been done. In theory, anything is possible, but very unlikely because with CRISPR you are taking out a big piece, of the gene. So hybrids usually happen in sandflies. So there is the potential of having a hybrid in a host is very, very low because the host does not have those conditions for hybrids to exchange the genetic material. You need the sandfly microenvironment. Okay. That has been shown by David Sachs very clearly. And so that's one thing. And so I think it's very low risk. And even if, let's say that's why we are using a dermotropic strain. So let's say there is some pathology, we can zap it with a radio frequency. We have a treatment, which is a topical heat therapy, which is WHO approved, which is like a 1 minute we can zap it. And a third reason why we use it is because as I said, that this is a dermotropic species. This is not visceralized. So basically you will see it in the skin and you can treat it very effectively. And 90% people will self-resolve anyway.
Okay.
That was it.
All right, thank you so much. And thank you so much for your presentation.
Yeah.
And with that, We will go ahead and switch over to Dr. Rafael Vieira.
Yeah, so I may have to leave, Laura, because as I said, I have a meeting, another one.
Okay. Is it all right if we include your email in the follow-up with?
Yeah, sure. Yeah.
Okay, excellent. So
Okay.
if there's additional questions,
Yes,
you can just
please.
send them.
Okay.
All right.
Thank you.
Thank you so much.
Thank you.
All right. All right. So Dr. Vieira, or actually, Dr. Satoskar, if you could stop sharing your screen.
Okay, I'm going to.
Perfect. Okay, and then Dr. Vieira, if you could go ahead and share your screen, you may begin. Great, we can see it. Although we cannot hear you. That's strange.
Yeah, he has been saying to me that he has faced some technical problems on the audio.
Okay.
There is an arrow next to the microphone icon, which you can look at to the settings. Maybe it's connected directly to the computer audio rather than to the mic.
Oh, yeah.
He on Dr. Rafael's side. Or if you just unplug the headset, you might be able to voice through directly through the computer audio. No. Yeah, maybe.
I'll just give him a moment.
Yeah.
Hi, Laura. Working now?
Yes, we can hear
Yes,
you.
great.
Excellent.
Working?
Yep.
Yep.
Yeah, we can hear you now. You can put your
He's not
slides.
hearing us, I guess.
Hello?
We can just text him that
Yeah, I'll let him.
let him know he can go on.
It's working.
Can you hear this, Alan?
Yes, we can hear you. Can you hear us? Yeah, we can hear you.
Working?
Yeah, you can share your slide. Yeah.
Yeah, I don't have a return, so you can type on the WhatsApp, okay?
Yeah. Okay.
Okay, thank you.
Yeah. Okay. Okay.
Now I can see you.
I'm so sorry.
You're muted. He's muted.
Oh, we could hear you, and now it cut back out.
His mic is muted. Just unmuting you. Yeah.
I already turned it off. Yeah.
And now we can hear.
Okay. So let's go back.
we can hear you now.
I will start again. Okay. Sorry about the time and.
Yes, okay, you can show the slides.
Thank you. Okay, now you can see the slides and
Yep.
and
Yes,
working.
yes.
Okay. I don't have a return of the sound, so I don't know what is going on with but I can, if I can, I'm not able to listen the questions. I can put on the speaker of my cell phone and listen the questions. Okay.
Okay.
Okay. Let's go. Okay. Sorry again. Internet connections problem. So I would like to thanks again for the invitation. I think now that you are all seeing the slides and hearing my voice. So I would like to present some ongoing studies that we are developing here and in Somalia with ticks and tick-borne disease. So my university, the Federal University of Paraná, is located on the southern Brazil, which is composed by 3 states, Paraná, Santa Catarina, and Rio Grande do Sul states. And Curitiba is the capital of the state. So Curitiba is the biggest city of Brazil with an estimated population of 2 million. And when we consider the metropolitan area, we are around 3.2 million people. So the city is, it's an urban city, actually, but is full of parks and green areas all around. So people used to go for these parks to walk and to rest and play with the kids. So Curitiba is considered a green city in Brazil, and we have a lot of animal species sharing the same environment, and that's why I'm working with ticks, because the parks are full of ticks here. So Curitiba is a sister city of Columbus. That's why it's a good way to make a partnership with Ohio State. And we just received a visit from Dr. Roy Dixon and Dr. Latz on February, along with Jane Aparecido, in order to make strategies to increase the collaborative studies between Ohio State and UFPR, particularly with GOHi. So this is our veterinary teaching hospital. Here we received animals from all the states, and where is located my lab, which is the Vector-Borne disease lab. So I will just show the currently funded project. We have 2 main lines. One is wildlife, which we are working with capybaras and opossums. as a urban wildlife, and another project with the roadkill animals, and another one using domestic animals. They are not until now they are not zoonotic pathogens, but we discover a new relic species infecting horses, and we are also studying small ruminants on the epidemiology of Anaplasma marginale. So the first thing that we started was to map where Brazilian spotted fever cases were occurring in the state. So Brazilian spotted fever is the most deadliest disease in the world. And we are close to São Paulo, which is the state with the highest prevalence and incidence in general of the disease. So we have until now 56 cases with only 5 deaths, which is located on the northern part. You can see on the map. But we have only 50 cases of human parasitism by ticks. So the first questions that we want to answer on this paper that is already being considered for publication at Ticks and Tick-Borne Disease Journal was why we have so many cases and the health state department does not report tick bites on humans. So we go ahead and we follow since 1993 the reports of tick bites on humans in order to know which are the most common tick species that are biting humans here, to really try to measure the risk of being infected by any Rickettsia species. It is surprising that when you see the NP on the map, this region is where all deaths were recorded, but we don't have any information about the tick species that are biting humans. And this is clearly a lack of reporting. And that is now we are working along with the state health department in order to prepare and build capacity on the health workers to really work on site when case happening and try to make a real record of cases. We also gather all reports of ticks occurrence on the state. We gather data from 1953. We have 2 scientific collections of ticks here on the state, and also we do very good literature review, gathering all publications with ticks occurrence or reports and tick-borne disease in order to map and see where are the ticks, where are the main species that are responsible for Rickettsia transmission, in order to make a program to help the health state department to elaborate control measures to avoid new case and new deaths. So the main tick species that are responsible for Rickettsia transmission here is Ambiomys sculptor, as you can see on the red stars. So these are the places that we have occurrence. And then other areas that are all, that you cannot see the circle, triangle, or anyone. There are, is not that we don't have cases, we just don't have reports or any information. And that is where now we are focused our efforts to know. So after gather all these data, we are working with the state health department to train and build capacity on health workers and all and trying to show the decision makers that we need a really good effort in environmental searching for ticks. So we are training them for dragging and flagging techniques, and also carbon dioxide trapping. We are also working with the people that are working with dengue fever in Brazil, the entomologist guys, and try to combine when they are going to the field to look for sandflies. We are also sending people to look for ticks. And then they all have a 1 week training section inside the lab. We go to the field, we sample the ticks, and then we get back to the lab. And my group is training them to at least identify the 4 most important tick species in order so they can have a faster answer in case of any suspicion of cases. But considering Brazil that I'm sorry about our president, I think he is very famous because of his mouth. So, but Brasilia now is facing a very bad science moment without any funding. And to make this Rickettsia Brazilian spotted fever project going on, we are working with side projects in order to get funded. So the first one that is really helping with money is a project that I approved with the clinical and molecular characterization of a new hemoplasmic species in capybaras. So this project is important because capybaras are everywhere here. So they are really in close contact with people. You can see the capybaras and cars, and people are walking, jogging, doing picnic with the kids. So they are really sharing a same environment, and we need to see what is happening with these big rodents. During this project, we have good side data. The one I would like to share with you is the one that was awarded with the best poster presentation, my PhD student on the last ICOFA, which was the potentially novel species that we discovered in capybaras. This study is part of his PhD and is already submitted for the journal. And during the work on capybaras, we also noticed that we have opossums everywhere. And these opossums, different from capybaras that rely on the river regions because they used to regulate their temperature with the river, the opossums really spread the city, and it is a good model for spreading ticks as well as spreading other potential zoonotic pathogens. So we also use wire net traps. And just to know, we have Iguazu Falls. I don't know, I think you are all know about Iguazu Falls. Iguazu Falls is located on the border between, you have on the left Argentina and Paraguay and Brazil, and you have a beautiful waterfalls there. They are competing with Niagara just to know which one is the beautiful ones. But it is the second place in Brazil in number of flights. So it is a really important and key city in Brazil. So particularly on this city, we have spread 70 wire mesh traps in order to capture opossums and do very good screening on zoonotic pathogens as well as on ticks occurring on those species to really try to evaluate what is the risk of zoonotic transmissions between opossums and humans and people that live on the city. Also during this data, we also discovered a new species of hemotropic mycoplasma. This data was already published, and now we just finished the whole characterization, and we are proposing a name for this pathogen, which is already also being considered for publication. When I was last year, I visited GOHi, and I was also discussing with Laura. Laura worked with Ethiopia, did a very good work on rabies, and we started to do a study here in Brazil with roadkill animals. Here we have two main species that are roadkilled. One is opossums that we are working on the urban areas, and another one is the porcupines. So we built a project on free living and roadkills, wild animal in general, but we really received opossums and these little guys, porcupines. During this project, in fact, it is surprising that we discovered another new species of hemotrophic mycoplasmas in these guys. And it seems that this wild, new, and potentially native species of hemoplasma does not cause any clinical disease on the animals. All animals, at least until now, the other one, wild canids, wild phthalates that we captured and sampled, they are really highly infected, a high parasitemia, but no any clinical signs, even anemia. So we are trying to work on that now. And then we arrive in Somalia. So Somalia, for the guys that are from Africa, they are really know about the country. It's a country that currently, since the 90s, the civil war has destructed all the education and health systems, and they are trying to increase their quality of life, let's say like that. And during the ICOFE in 2017, I was fortunate to know Dr. Abdallah Ibrahim, which is a faculty there. And we did a very good meeting, and we started to do some combined projects. So since then, I'm trying to help those guys to improve science, mainly the quality of life for the people there. So we had built an agreement between Abhay University and Federal University of Paraná, and I'm also in depth with GOHi, particularly Dr. Gebrais and all folks that are helping us in trying to build some studies. And we have designed some initial studies to screen animals and screen people to at least know what is happening there. So we can later try to organize and elaborate any control or any other measure to try to impact on the good side of the animal production. So for these, we are capacity building some people from there. The first one that came, it was Dr. Ahmed Hassan, which is performing his PhD here with me. Later, we received Dr. Abdul Karim Yusuf. He's pursuing his master's with me, both working with Vector-Borne in general. Dr. Abdallah was intending to come this year, but due to the pandemic, he postponed his coming for 2021. And we are also receiving some undergrad students to perform the last part of the course, and then they can be trained on PCR methods and ticks identification as well. So as on the broad picture, we are working in trying to help them on the new veterinary medicine curriculum. We are doing some transversal courses on a week basis. We have also webinars through our groups. And also I'm teaching some graduate courses for them on epidemiology of vector-borne disease, morphology and biology of ticks, development of epidemiological studies, and some basic notions about molecular biology diagnostics. These are some pictures from the Abhay University that they share. We worked on choosing a good stereo microscope for them to analyze the ticks, so they can really evaluate on high resolution, and the ones that they are not use it, they can just take a picture and send me through email, and then we can follow and improve the research. So the ongoing study is molecular biology of tick-borne disease in animals and humans from, particularly from Mogadishu, which is the capital of the country. This study until now is not funded by any agency directly. We have UFPR and Abhay University funding part of the student of the research, but we also have the two main agencies of Brazil, which is CAPES and CNPq, which are providing the full scholarship for the PhDs and master's students. The ongoing projects that we are running now, we are evaluating sheep, goats, and the owners under Mogadishu city. We are also working on the camels and the workers. Somalia has one of the biggest dromedary of the world. So it is a quite important species to evaluate. We are also trying to evaluate some occupational disease on vet students and non-vet students from the Abhay University, and also started to work with immunocompromised patients like HIV positive, cancer patients, and pregnant women in order at this first step, screen and to know what is happening first, and then we can elaborate another proposal. We have a lot of ticks already sampled. We already did on 2018 the first sampling. All samples are already here. Dr. Ahmed and Abdul Karim get back to Somalia to do another sampling. They were supposed to come back on March, but now we don't know when they will come due to COVID-19. But we already have some side data that we are working and publishing on the journals. These are some very good pictures that they took from this first step. We already did some publications. This one was already presented on GOHi last year. At the end of last year was published on Parasites and Vectors Journal, which was the first description of molecular diagnostic of trypanosomes in camels from Somalia. We characterized Trypanosome evansi and Simia in camel, and we all work with nomadic and dairy farms, and we found a higher prevalence on nomadic animals. We just submitted another one with cattle, goats, and sheep. And this one, we worked in two different districts, which were Afegoy and GOHi. And we found a very good data when we found that in GOHi district, the prevalence in cattle was really lower than in Afegoy. And going deep on the epidemiology, we've noticed that the GOHi people used to graze cattle during the night, which decreased the odds of being bitten by the tsetse sunflies. So I would like to thank, due to the very short time, I would like to really present a broad range of what we are doing. Now we are needing partners to join our group, and we have a lot to study, which is not my area, but samples are here and free to be used by any group. Rabies, toxoplasma, and other very important zoonotic pathogens. I think it's what I had to present now. Thank you.
Thank you so much, Dr. Vieira, for your wonderful presentation.
Hearing, I don't know, I'm able to questions. I think
yes. So
could call me through WhatsApp, so I can, or you can type here and I try to answer. Yeah, the audio is not working. Okay, get that. Thanks, Desalene. We had a really good discussion in Quebec about this project, so I'm pretty excited and looking forward to spread the study. While we are so we have a quick question. Do you know the species in Somalia? So good question, Laura. What we have now? We mainly found two main tick species on goats, sheep and goats, which were Rhipcephalus pulchellus and Rhipcephalus lumeralis. We are now desiccating because we noticed that people used to PCR the whole tick. But since tick were attached to the animals, we cannot say that this tick was PCR positive or not. So we are desiccating and we will PCR the salivary glands in order to know which pathogens. We have screened some samples. Firstly to hemotropic mycoplasmas, but we have some good data on Ehrlichia and pyroplasmas that we have initial sequences, but since COVID has closed all labs, we have some amplicons here that we need to send for sequencing in order to know the species. It was really on the beginning of the study, so that's what we know now. We have another. We have also Yaloma dromedaria on camels, and we have some argazide that we collect from humans. So, but pathogens, nothing until now. I hope I answered your question. So, guess not.
Yeah.
Good. So, GOHi, the main thing now is about tick species. We have in South America, we have a totally different tick species from Somalia, from the whole Africa in general. Here we just have two Rhipcephalus species, which are Rhipcephalus micropulus, which is worldwide spread, and Rhipcephalus sanguineus. We have two lineages in Brazil. My area has mainly the tropical one, which is responsible for Ehrlichia canis transmission, but this lineage does not have any anthropophilic behavior, so the humans are quite safe. But in Somalia, we are dealing with different species from South America, but the prevalence in general are not differing from like data that I have found for Egypt, from South Africa or other countries. I think the most important is that to know what is happening in Somalia, do this totally isolation, security problems. So I was supposed to go there on 2018 and I was not able. So actually we had a talk about that. So the first thing I think is to know what is happening there and try to build an agenda to help them. So here, what we have, the another one in Brazil, what we are trying to do. So we have two different situations here when we compare to São Paulo state, which is the most important for Brazilian spotted fever. So São Paulo state is a dry state. It is on the Cerrado biome, and Paraná here, at least Curitiba, is the highest capital of Brazil. So it is dry, it is cold and humid, and we are on the middle of the Atlantic rainforest biome. So the Amblyomys sculptum, which is the main tick species, is not able to live here. So we have another tick species that might be involved on the epidemiological cycle. So now we are trying to map where are the ticks, and then the areas that they have a high level of infestations, we try to put signals in order to people avoid that places to make a picnic or like trails to walk or run on that place. It is at the moment what we are doing, and we are also doing a small book to work with the kids on environmental education, showing them that if that park is infested by ticks, that they avoid that area to walk with the dogs, or in order to avoid to walk with the dog there, and then the dog will get infested by ticks and then go back to home and bring the ticks that might be infected by Rickettsia species and put them on a risk. It's basically what we are doing, building capacity of the health workers. In general, all money for vector-borne disease in Brazil goes to dengue fever. So guys that are working with Leishmania, like Dr. Satoskar, and we as a tick-borne disease, we have to handle the health workers to give some time to look for ticks or other vectors other than Aedes aegypti. I think I missed that. Yes, perfect. Perfect. Thank you for your time. Sorry again about this internet connection and sound problem, and I'm open to answer any email questions that you follow up. Stay safe, everybody. Take care.
All right, thank you so much for joining us. Thank you, Dr. Vieira and Dr. Satoskar, for your wonderful presentation. Our next webinar will be June 18th, and we'll be sending out the summer schedule shortly. Thank you so much for joining us, and stay safe. Bye.